Welcome, everyone. Thank you for joining us at the Tissue-Based Research Workflows virtual event. Our next presentation for today is selecting the right detection for your IHC ISH project, Fluorescence versus Chromogenic Staining, presented by Shenna Washington. My name is Serena, and I'll be your moderator for today's event. Before we begin, I'd like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want during the presentation. To ask a question, click on the Q&A box located on the bottom of your screen. Type your questions and hit submit. The speaker will answer your question following the presentation. If you have trouble seeing or hearing the presentation, please click on the Q&A box to let us know that you're experiencing a problem. Please join me by welcoming Shenna. For a complete biography on our speaker, visit the Biography tab on the left. Shenna, over to you.
Hello, and welcome to the Leica Biosystems presentation on why quality matters, and my little segment on selecting the right detection method for your IHC and ISH assays. So my name is Shenna Washington, and I am a Senior Field Application Specialist in Advanced Staining for Life Science with Leica Biosystems. I've been with Leica for 5.5 years. Previous to that, I worked in research histology for approximately 17 years, doing IHC, IF, standard staining, microscopy, fluorescent microscopy, confocal microscopy, all kinds of different things for both human and animal tissues. And I've worked in academia and CROs and now in the commercial industry. So a little bit about me. So I want to clarify before we get into the meat of this presentation that the presentation does assume some basic knowledge of IHC and ISH concepts. It's basically antibodies, antigens, probes, RNA, DNA relationships, heat and enzyme antigen retrievals, antibody titrations, dilutions, things like that. So to gain the most from this information covered in this presentation, you will need some prior background and experience with IHC. So we will jump into it here. So, summary of this is how to select the most appropriate detection method for your work. So some initial considerations we'll talk about first. Chromogenic versus fluorescent labeling. We'll move into single versus multiplex labeling, which everybody gets very excited about, and available technologies, and finally some best practices. So choosing the appropriate detection method for your targets will allow researchers to produce the most compelling visual images to support their research goals. We'll discuss some of the major factors that contribute to effective single and multiplex stainings with focus, of course, on quality. And then we'll talk about some best practices for assay development. So talking about initial considerations, these are going to be some key concepts you want to think about before you purchase anything, before you spend any money. So what are the research questions that I need to answer? First and foremost, that's what you need to be thinking about. The desired endpoint typically determines what kind of detection method will work best for you. So ask yourself, what is it that you really need to demonstrate? You know, there are lots of fun things out there. It may be a fun idea to put 20 markers on the same piece of tissue, but do you really need that? And in the end, are you going to be able to analyze that effectively? Next, good question is to know what's already in your lab that you have at your disposal. So what kind of microscopes do you have access to? What kind of reagents do you already have in-house? That leads directly into the next question of how much time and how much money do I have? So what is your budget for the project? Can you afford to try something new? What is the timeline for the project? And do we have time to develop something completely new? So those are big considerations. You don't want to waste, most people don't want to waste a lot of time and money. The next one is who will actually be doing the work? So do we have the manpower to develop a new assay? Do we have personnel with the skill and experience to develop that assay? So those are some big considerations too. That leads into how will you analyze the data? Do you have the right analysis methods for the type of detection method that you are selecting? So if you have, obviously, if you've only got light microscopes in-house, a fluorescence detection assay is probably going to be pretty pricey because you will also have to find a fluorescent microscope to use somewhere or buy a whole new one with all the different filters and different things that go along with that too. So if you've only got light microscopy in-house, you may want to go with a bright field chromogenic detection assay. So do you, who will analyze the data? So that's always fun to know. Like, will it be student helpers doing the analysis of the data? Will it be a pathologist doing the analysis of the data? So knowing who these final slides are going to be read and analyzed will be key in making it useful as well. So another big consideration is what kind of support do you have access to? So do we have access to resources to support our potential knowledge gaps? Do we have access to resources to assist with any issues that may arise with developing the assay or analyzing the assay, the outcome of the slides? So those are some really important things to think of before you even get started with purchasing the newest, latest, and greatest, funnest detection methods out there on the market right now. So now we'll get into the differences between the two main detections we're going to talk about today. So the chromogenic staining. So chromogenic detection assays, they first and foremost are the predominant detection technology in clinical settings, and primarily it's just like what we're looking at right here in this photo, the DAB diaminobenzene substrate with its familiar brown color. So this is still widely used in research immunohistochemistry as well for assay development, proof of concept, nothing else. The nice thing about this, like I said, it's widely accepted. So acceptance is the #1 top benefit of this. People know how to read DAB stains. It's widely accepted. They know what they're looking at. Second is the cost. It's relatively inexpensive detection option. There are many different options out there, so availability is another. There's a range of different enzymatic detection. Commercial vendors commonly use the DAB test to demonstrate their IHC products and antibodies. So lots of options out there, highly available. Microscopy. It is a highly accessible detection technology as it can be visualized by using standard bright field light microscopes, which most labs already have available to them. So the range of substrates and chromogen colors, that's also a benefit. The DAB substrate works with horseradish peroxidase enzyme, which is a robust enzyme, now has several color substrates. Other than the standard DAB, you can do yellows and blues and greens and purples and reds. There are also a couple of other enzyme options for detection. The one we see most often is the AP, the alkaline phosphatase enzyme. The AP detection also offers a range of different color substrates, red, blues, greens, etc. All right. So fluorescent detection is not as widely used and accepted as the chromogenic detection methods. It has been growing in popularity, and we do see it quite often in research. And there are just as many fluorescent detection reagents and options out there. But most of the time we see people choosing fluorescent detection out of necessity because the options for direct labeling is one major reason. This can be fast and efficient for optimizing and visualizing certain markers. Direct detection often requires less development and skill to produce high quality results. So if you've got less experienced people working in the lab, this may be a good option for you to take. So alternatively, indirect labeling is another method you can use both for chromogenic and fluorescent detection. But alternatively, it introduces more variables, but it also provides a bit more flexibility in the sense that you can use the same primaries or probes with different colors. So you can mix and match a bit more. But direct labeling is a nice option there with the fluorescence. The most common reason we see groups choosing fluorescence in research right now is because they are looking for multiple markers that co-localize. And by co-localize, I mean they present in the same cell or the same cellular compartment. And when chromogens overlap, it can be very difficult to visualize and analyze the staining results. And this is not really an issue for fluorescent labeling. So as long as you are choosing fluorophores that are far enough apart on the emission spectrum, you can easily visualize and distinguish between different targets on the same cells with fluorescent detection. Fluorescent detection technologies also offer the ability to detect more targets on the same sample. So like I said, if you've got co-localizing things, you don't have to worry about them overlapping. So we very often will see, 7 or more targets being labeled on the same. Not all of them overlapping, mind you. It's still be quite a challenge to try to see 7 targets overlapped in the same space, but you can get 7 and beyond targets on the same tissue sample on the same slide. So that has become a very huge benefit of fluorescent detection. Recent advancements in imaging technologies have made visualization of both chromogenic and fluorescent much easier than it used to be. So it's not always a huge concern, but it's something to keep in mind. Multiplexing considerations. So if you are looking to detect more than one marker on the same piece of tissue, so got a couple colorful photos there, demonstrations of multiplexing. So any kind of multiplexing assay requires a solid comprehension of the basics of basic IHC, basic ISH, and automated platform if you are using one. So if you're using an automated stainer, you need to have a solid comprehension of its function as well, what it's capable of doing or not. So with any multiplex assay, there are many variables that can require intensive development work. So tissue-based multiplexing assays can be as complicated as they look here. So you look at this and it's a little boggling to the eye, and it can be just as complicated to try to get a beautiful outcome like this. There are many variables to consider, so it's important to start with those fundamentals. Primary antibody compatibility is a primary concern. Then we've got optimizing your antibody or probe titration, optimizing your pretreatment conditions, chromogenic and fluorophore efficiency, changes in signal through subsequent processing. So we will take those one at a time here. So primary antibody and primary probe compatibility. So these things, primary antibody quality, and when I say quality, I'm talking about things like affinity, which means the specificity for a particular antigen. Avidity is another concept, so strength of the binding to the target, and then prevalence within the tissue. So those are some things to keep in mind when we're talking about antibody quality. Lots of choices out there commercially available as far as antibodies. Can be challenges when we've got antibodies that are generated in-house, but quality is certainly a big concern. Species is another concern. So the host species for your primary. Mostly we think about cross-reactivity, which can occur when antibodies from the same host species are being put onto the same tissue. And then the other thing we worry about is cellular location, where we can have issues of steric hindrance or just hindrance of binding due to the previous antibody that's been placed on, or even just obstructive deposits left by substrates, especially if you're working with chromogens. So those are some concerns with your primaries. Depending on the technology you choose, this may or may not have a major factor, be a major factor and have a major impact on your assay development. Some technologies are made to be species agnostic, and ISH probes can be very specific to sequence. So the success of other detection methods may depend heavily on compatibility of antibodies and targets, clones or sequences, but some things will definitely be species agnostic. So that's just a couple considerations with your primaries. Next thing, optimizing your antibodies and probe concentrations. So this is key. Certainly, if you are going to, like I said earlier, try to throw 20 markers at a piece of tissue at the same time, you're going to need to know the optimized dilution concentration of those primaries and the probes as well. So people often attempt, right out of the box to throw all of the desired markers onto a piece of tissue at once, which can provide some baseline information, but rarely produces the desired quality of outcome. So optimizing the individual antibodies or probes prior to combining them will provide a good sound baseline for understanding the expected performance of each marker. And it will aid in the decision-making process for tweaks that may need to be made later in the assay building process. So once the markers are combined. Optimizing pretreatment conditions. So once you've got you're working out your optimized titration for your antibodies or probes, you will also want to work out your optimized conditions for pretreatment. So each marker in a multiplexed assay may require a different pretreatment to produce the best quality staining results on its own. So whether this is higher, the heat epitope retrieval, or enzymatic retrieval, it'll be crucial to determine this for each marker. So in order to determine the retrieval conditions necessary to incorporate into the multiplex assay. Chromogen and fluorophore efficiency. So reagent solution stability is the first thing I think of when I talk about efficiency. So it refers to the general performance of qualities of the chromogenic substrates or the fluorescent dyes. Some color substrates or fluors may be much more robust than others. So for chromogens, when we look at stability in terms of the reagent itself, so once solutions are mixed, we can see precipitates. It may be sensitive to temperature or light. And whether the label deposit that you get from the final product can hold up to high temperature, to changes in pH, to salinity changes, and whether it is solvent resistant in the end. So once you've actually got the slides stained and you are going to mount them permanently or with aqueous mounting medium, some different chromogens, they may vary in the mounting needs. So fluorophores, we may have some that are always much stronger than others, and when we see a loss of signal due to photo bleaching over time, that's the primary consideration with the fluors. Potential changes in signal through processing. So as we are going through multiplexing and different rounds of different solutions going onto the piece of tissue, onto a slide, are we seeing changes with subsequent rounds? So we only really need to consider this most of the time with sequential assays. So if you are doing a parallel multiplexing assay, this is not going to be such a huge concern. Changes to staining over the course of subsequent processing with heat, with different pH solutions, different salinities, can really change the end of staining. So this is common with sequential, but a bit less with parallel. The effect makes determining the optimal order of application for your markers and their associated staining conditions, so epitope retrieval, stripping methods, makes it crucial to the quality of the final staining result. So chromogenic multiplexing stains, here's a little example. This comes from my own portfolio and my own experience. I couldn't tell you exactly what the markers are anymore. I can't remember, and did not have it labeled on the photo. So, but I know that this is a CD marker. We've got probably a FOXP3 or TBET and a KI67, I believe, at the time, or Bond RX. So this took, let me think, this took me probably a few months in development. And these markers, this is on a human tonsil, so this was just my test run here. The image was also produced on a Leica light microscope. So considerations here were the limitations of the automated platform at the time. Is it capable of doing multiplex chromogenic write-off? Does it have features that lend itself to that? Solution stability was a huge issue here. So you kind of see that blue-green color. That chromogen was not stable over time. It had a life once mixed. Once it was mixed, it was labile, and it would start to precipitate out of solution after about 15 minutes. So we had to keep that in mind. Cellular location. So you can see here that the brown color is very ubiquitous all over the place in there. So we wanted to put that one on first. The DAB was a more stable chromogen and held up better through subsequent rounds of staining. So we put that DAB brown color on, and it did take some balancing. So we did have to back off on concentration to get it to be that nice light brown so that you could get the red color to kind of show through it. And same thing with the blue-green. So we wanted to balance the colors, so that took some tweaking of the antibody concentrations and the epitope retrieval conditions. Determining cross-reactivity, it's always nice to be able to do some testing with your chromogens and stuff. So you'll work them up one at a time, and then you'll work them up in combination. And there are some tests you can do to determine whether you're getting cross-reactivity or getting a real signal with multiple markers. And then visual contrast compatibility. You can kind of see by looking at this that the brown and the red are very close in the color family. So can be tough to distinguish red from brown, and especially if red and brown are on the same cell, if they're co-localized, can be very tough to see this. Whereas the blue-green has a nice strong contrast against those colors. So I had to keep that in mind as well. And then order of staining, kind of discussed that already, where we decided to put on the CD marker, the very ubiquitous brown first, because it held up better through subsequent rounds of staining. I believe then the red went on, and then we had the blue-green in the end. So moving on to a fluorescent multiplex. So this, I believe, is exactly the same assay, exactly the same markers on a human tonsil, but this was done with fluorescent detection reagents. So I developed the IHC assay first, and then I tried to move it into a multiplex immunofluorescent assay. And that really sped up the process, having the whole thing worked out in chromogens first. And there were some processes involved, that were quicker with the IF. The chromogenic development was not a consideration, so some things went a little more smoothly, but in the entirety of the development, it still took about a month to transition to this IF assay from the original IHC assay. The microscopy and imaging was a challenge at the time. All we had in the lab that could do the fluorescence analysis and detection was a confocal microscope, so it was a Leica confocal. And then the biggest challenge for me at the time was that we, in the lab, did not have a good way of analyzing this data. So we did not have software in the lab that we could use to separate out all and do cell counts for us or anything like that. So after months and months and months of doing all this multiplex detection work, both the chromogenic and the fluorescent, it sat on a shelf and wasn't able to be read right away because we did not have the analysis in-house. So available technologies, there are lots of things out there. When we talk about enzymes, we talked about the prevalence of the HRP, the horseradish peroxidase enzyme, and then the alkaline phosphatase enzyme. We see those pretty regularly. There's a beta-gal, the beta-galactosidase. That one is a little bit more obscure. Amplification. So a lot of people want to see nice dark or nice bright staining of their markers. So they like to try to amplify the signal. Polymers, oligos, TSA, haptens, lots of stuff out there on the market. So break-apart technologies are another new thing that are allowing spatial relations and quantification. So there are different technologies out for that as well. And then service labs can be a really great option for those who do not have the resources to develop their own assays in-house. There are lots of groups out there now who will do the staining as a service and as well as the analysis as a service. So as far as best practices go, early inventory of needs and resources is crucial. It's always good to take stock of what you have at your disposal and then a good hard look at what you actually need. Review of reagent specifications. Make sure that you know what you need to work with your reagents. So know the stability of your reagents, know what they are compatible with and not compatible with. And once you know that, go back and take stock of what you already have and what you don't have and what you may need. Specifically, a specific example of that would be chromogen substrates. So some of the substrates are not solvent stable or solvent proof. They will dissolve in different organic solvents. So you can't run them down the way you normally would just a DAB stain or something like that. You may get some products in the end with absolutely no staining on them because it dissolved into your mounting media. Automated platform compatibility. So there are lots of automated platforms out there right now. They have different capabilities, different specialties, pros and cons about all of them. A lot of people will decide they want to do something and then find out that their instrument actually cannot do that fully automated or cannot do it at all. So it's good to know the actual limitations of your automated platform if that's the route you're going to go or that you need to go. And then the final one, absolutely hands down, being diligent development. So optimizing things one thing at a time, good scientific method, and adjustment of just one variable at a time as you're going along will produce the best quality outcome. So just some good references here. An author that really helped me when I was going after information on this and doing it myself in the laboratory, Chris Vanderloos. So a couple different publications here of his. And that will end my presentation. I would like to thank my life science applications team and my mentors. And thank you. We'll take some time for some questions.
Well, thank you so much, Shenna, for that informative presentation. Just a quick reminder to our audience on how to submit questions. Simply type them into the Q&A. We will answer many questions as we have time for. And the first question is, how do you know if the chromogens you want to use in a chromogenic multiplex are compatible?
So chromogens are very dependent on the vendor that they come from, right? So it's very important when you're choosing chromogens to check the data sheet and the instructions for use, to know what solutions they may or may not be compatible with, to check whether they are solvent stable, so whether you are going to be able to cover slip them the way you normally cover slip things in your laboratory. So check the data sheets before you go picking out and ordering certain things, because very often different chromogens may or may not be compatible. So specifically look for solvent stability. So can you run them through graded alcohols? Can you run them through xylene? And then check whether there is any listing of the kind of mounting media they are compatible with. So are you able to use permanent mountants that contain xylene or toluene or things like that? Or are they going to require aqueous mounting media? And if they are not compatible, if you've got two, one that requires aqueous mounting and one that requires permanent mounting, you may end up with only one color on your slides in the end. So very important to check the data sheets and instructions for use on your chromogens.
Very good. It looks like we have one more, unless some more come in. The second question is, how do you know whether your automated platform is suitable to perform the kind of detection you've chosen?
Automated platforms can vary a lot, so that's very important. Again, I talked a little bit about this in the presentation, about knowing the functionality, knowing the limitations of your automated system. So things like, is it able to mix solutions? So most of the chromogens that you order come as component parts. So they're modular. They come with one, two, or more solutions that you're going to need to mix together to apply to get the substrate that you need on the slide. So can your instrument actually do that, or is that something you're going to have to do offline? Is that something you're going to have to do by hand? So can keep that in mind. Another thing would be checking the protocol. So if you have a protocol that you've gotten from literature, or that is listed with the technology that you are considering, make sure that your instrument is capable of doing the things that are needed in that protocol. So is it going to require heat? Heat is a big one. Can your instrument heat within the protocol that you are trying to use? So I would say those are the two biggest concerns. Mixing of solutions, is your instrument capable of mixing solutions, and also heat being included in the assay that you're running. Is it able to heat?
Yeah. Fantastic. Well, thank you again, Shenna, and thank you to our audience for joining us today and their interesting questions. We will address any questions we did not have time for today, and those submitted during our on-demand period via the contact information you provided at the time of registration. This session webcast will be available on demand after the event. Thanks for the session, and thanks for joining us.
Thank you.
Bye for now.